Hello everyone, I am Jithma Prasad and I will be presenting our publication on behalf of all my co-authors. The title of this manuscript is Cyclin D1 overexpression induces replication stress and microhomology-mediated end-joining dependence in mantle cell lymphoma. This is the visual abstract. In this study, we have shown that increased cyclin D1 expression causes replication stress, which leads to physiological DNA double-strand break accumulating in mitosis, thus increasing cellular dependence on a mitotic-specific double-strand break repair mechanism. MMEJ is the main double-strand break repair mechanism in mitosis. We have shown that targeting MMEJ can cause significant antitumor activity in cyclin D1 overexpressing cells. This dependence is further increased with concomitant ATM deficiency as it further increases replication stress-associated double-strand breaks. First, we overexpressed cyclin D1 in U2OS cells and showed that increased cyclin D1 expression causes significant replication stress and DNA damage. Profiling double-strand break repair mechanisms or double-strand break repair pathways showed cyclin D1 overexpression significantly increases MMEJ activity without much alterations in non-homologous end-joining and homologous recombination repair pathways. This increased MMEJ activity correlated with increased expression of polymerase theta, the primary polymerase of this pathway. We used mantle cell lymphoma as the model to study replication stress associated with cyclin D1 overexpression, as mantle cell lymphoma has universal overexpression of cyclin D1 due to 11:14 translocation. In mantle cell lymphoma cells, we profiled physiologic DNA damage and showed that in the absence of polyQ, significant DNA damage accumulates in the M phase of the cell cycle. Further, polyQ absence also significantly increased replication stress in addition to mitotic DNA damage. Correlating with this mitotic DNA damage, absence of polymerase theta increases chromosomal aberrations as depicted by increased prevalence of double minute or dicentric chromosomes. Further, the absence of polyQ also increased single-strand DNA in mantle cell lymphoma, another characteristic of replication stress. The upregulation of polyQ by cyclin D1 overexpression is specific, as we show by CRISPR interference mediated decrease of cyclin D1 expression, decreased replication stress, and DNA damage markers along with MMEJ dependency. The results were not confounded by the effect on cell cycle by this controlled decreased cyclin D1 expression, as the level of decreased cyclin D1 did not cause cell cycle arrest. Mechanistically, we show that cyclin D1 binds to polyQ promoter and causes activation of polyQ transcription. ATM is the second most common genetic alteration in mantle cell lymphoma, seen in about 40% of cases. We then ask the question, what happened to the wellness of replication stress and DNA damage repair mechanisms when ATM is deficient in cyclin D1 overexpressed background? We show that concomitant ATM deficiency in cyclin D1 overexpression further increases MMEJ activity without altering HR or NHEG activity. Correlating with this, we show that polyQ expression further increases with ATM deficiency in a cyclin D1 overexpressed background compared to cyclin D1 overexpression alone. Translating these findings into mantle cell lymphoma treatment, we show that polymerase theta depletion decreases mantle cell lymphoma proliferation and increases apoptosis. This phenomenon is further enhanced with concomitant depletion of ATM with polyQ. By using multiple mantle cell lymphoma cell lines, we show that targeting polyQ by novobisin or ART558 induces significant antitumor activity, and co-inhibition of ATM and polyQ is synergistic in vitro. By using a xenograft model, we show that drug-mediated polyQ inhibition decreases mantle cell lymphoma growth, and this antitumor effect was further increased in mantle cell lymphoma with ATM deficiency. Moreover, replication stress and DNA damage repair markers were significantly increased in tumors exposed to polyQ inhibition, a phenomenon that is further augmented in ATM-deficient syngeneic mantle cell lymphoma cells. We also validated our findings in primary mantle cell lymphoma patients. We show that primary mantle cell lymphoma cells have relatively high expression of polymerase theta compared to other non-Hodgkin lymphoma. ATM-deficient mantle cell lymphoma has relatively high expression of polymerase theta compared to ATM-proficient mantle cell lymphoma. Drug-mediated polyQ inhibition causes a significant anti-tumor activity or anti-tumor effect in primary mantle cell lymphoma cells, and ATM-deficient mantle cell lymphoma cells were more sensitive to polyQ inhibition compared to ATM-proficient mantle cell lymphoma cells. In summary, we show that Cyclin D1 overexpression increases replication stress and causes increased dependence on MMEJ-mediated double-strand break repair for cell survival. This replication stress is further increased with ATM deficiency, as is the MMEJ dependence. This biological dependence can be successfully exploited for therapeutic benefit in cancers with Cyclin D1 overexpression, such as mantle cell lymphoma, through polyQ inhibition. Thank you.